tutorial serves as a brief video introduction to the tool cellHarmony, which is a component of the software AltAnalyze. The goal of cellHarmony is to take two independent single-cell RNA sequencing datasets and compare them. Specifically, the program identifies cells from one dataset, for example a mutant, that map to a wild-type reference. Once those cells have been aligned, a differential expression analysis is performed to identify both genes that are differentially expressed within each cell type, differential population frequencies, and identify pathways that are impacted. To begin, you'll need the software AltAnalyze. So go to altanalyze.org if you haven't already. Download the software by simply clicking on one of these links. For example, I'm going to do this for Mac. It will take a minute to download, or PC. If you're doing this on Unix, we recommend using pip install AltAnalyze. You may need sudo pip install AltAnalyze. Once that's installed, you'll be able to call that on the command line simply by typing AltAnalyze. For example, AltAnalyze, if I can spell it right. There you go. So this calls up the graphical user interface, which you can begin from here. If you download the Mac or Windows installer, it's going to be a similar interface, or it's actually going to be the same interface. If you also are using the command line interface, you can use the command line options. You don't have to use the GUI. These are present on the bottom of the cellHarmony documentation, which we'll walk through in a second, using these kinds of commands. However, for the purpose of this tutorial, we're going to actually be walking through the graphical user interface. So first off, you'll want to go to the cellHarmony main page at AltAnalyze, which is altanalyze.org, cellHarmony. From here, there's a number of links, including the cellHarmony documentation, a video tutorial, the video tutorial we're watching right here, which would be crazy if we actually were able to click on this. The AltAnalyze manual, instructions and documentations. There's a simple R package that does the alignment steps, but not the differential expression analyses, and a Seurat R conversion code, which allows you to generate the AltAnalyze compatible heat map format files and expression files that run the cellHarmony steps. There are also a number of references here that have also been pre-built, which you can align to identify prior defined cell populations. Once you have downloaded, for example on a Mac, the AltAnalyze installer, you simply can open that and drag the AltAnalyze directory within it to your computer. Here, by default, it's suggesting the applications. Applications don't work necessarily well for all users, since you may not have access to modify the directories within applications. So it may be actually better to copy to your desktop or somewhere else on your computer. So you just drag this, for example, onto your desktop. I've already done this, so I've got a version of AltAnalyze over here. AltAnalyze, you simply double-click on the program directory and find the program icon, which is a green icon. The first time you open, you'll need to right-click or control-click on this and say Open. This is a program downloaded from the internet, so the program will want to authorize that it is okay to use. You will say, yes, I've opened this before, so it's not asking me. If you had problems, you'd simply select this alternative AltAnalyze icon. More information about running AltAnalyze can be found right here in this link. You select Begin Analysis and Continue, and typically you will be asked to download a species database. I can do that here by selecting Add New Database. You just select the species database of interest and proceed. It will download. Once complete, you simply select that species within the software and proceed. I'd like to note that there's other folders here, including a demo data directory, and this demo data directory has inputs that we can use for performing our demo analyses. I'm going to select mouse musculus. If this is 10x genomics aligned data, I simply select 10x genomics aligned data and follow the directions from there. Usually, if you have 10x genomics aligned data, you've already run cellHarmony and it's produced a matrix file, which you can use input. In this case, we're going to use a tab-delimited text file. Both types of files, a 10x genomics example and a tab-delimited text file, are provided in that demo directory. I'm going to say process expression file rather than process RNA-Seq reads, for example, using FASTQ reads. I'm not going to do that here right now. I'm going to select the file of interest. In this case, I'm going to drag this demo data down here, and then I'm going to go to cellHarmony. I'm going to go to this mouse bone marrow RNA-Seq dataset, and there's more information about it in this README here. I'm going to select the reference, the expression input directory, and this expression file. This is a tab-delimited expression text file. I'm going to select an output for this experiment, which I'm going to save to expression input and reference. You can save it really anywhere you want. And for now, don't worry about these other defaults. You can modify these later. These include statistical thresholds, options for pathway analysis and pathway visualization, and a lot of other neat comparisons that you can perform. Normally, this is empty here, these fields where it shows different groups. If you want to perform a differential comparison analysis between known populations, you need to populate these in advance manually or through an external text file with a specific format. But in this case, we're actually going to run an unsupervised analysis using this option up here, say that says Run ICGS. We're going to select Continue. We have these default options that are shown here. You can change these if you want to look back at the ICGS tutorials. Typically, these are fine. Most often, the thing that people want to modify is to include or remove cell cycle effects. So if you really want to remove cell cycle effects, select Stringent here or Conservative, and then Run Analysis. So I'm going to select Run Analysis, and we're going to get this black window that appears. And in about 5 to 10 minutes, this analysis should be complete. Once ICGS is completed running, you'll see the different outputs of the program, which include the final output shown here. These different files are found within the program output directory that the user designated. In this case, we have over a dozen cell populations that were identified by ICGS, along with cell type predictions that are shown on the left. These cell type predictions are derived through a gene set enrichment analysis of each one of these gene populations, with the p-value associated with that prediction shown on the bottom. So in this case, this bottom cluster was predicted to be myelocytes, whereas this top cluster was predicted to be dendritic cells. If we close this, we can actually quit the program as well. Go back to the demo directory, demo data directory, where this analysis was run, and you can see the various outputs that are produced in this reference directory. The final output of ICGS is saved in this ICGS NMF directory for ICGS version 2, which is the latest version of the software. If you double click on this directory, there's a UMAP projection of the different cell populations that were produced. There's the heat map that we saw displayed within ICGS. For example, if you double click on this marker genes file and open it in Excel, you'll see the top ranked genes for each cell population as annotated within the heat map. So this, the text file, specifically the final marker heat map text file, can be used as a reference for cellHarmony. Alternatively, there are files within this folder ICGS, which represent the intermediate outputs of this program. If you select any one of these text files, these can also be used as a reference for ICGS, or I'm sorry, cellHarmony. So to run cellHarmony, you're going to go back to Alt Analyze. You're going to open up Alt Analyze again, begin analysis, select your species of interest, continue, select additional analyses. And here we're going to select the option Cell Classification. There are two main file selection options at the beginning of this program. The first is the input data set for the cells which you want to classify against the reference. In this case, the files that I have which I want to classify are found in this input directory in the demo data folder. This is the AML file. This is a leukemia model which is described in that README file within that directory. And our reference here is going to be the ICGS output that we just reviewed. So if we go to References, ICGS NMF, and this final marker heat map. The program will identify the full expression file that's associated with this ICGS output, which it knows to find in the expression input directory, which has the prefix exp dot. The program always assigns that prefix to the file as it's running. And then there are a number of cellHarmony options, which you can see here. The first is to whether or not to perform a differential expression analysis between the compared groups. We wouldn't want to do this if we were comparing two data sets that were, for example, produced by two different single-cell RNA-Seq platforms, for example, 10x Genomics and DropSeq, or 10x Genomics and Fluidigm, or potentially were generated in different laboratories in which there were batch effects. However, in this case, that's not true. We can also align individual cells to the cluster centroids of each cell population or the individual cells. Typically, the individual cells are going to be more informative. However, when you're analyzing a data set with tens of thousands or tens of thousands of cells, this is too computationally intense, and so using the centroid option is a more conservative option, should give similar results. There's a default correlation threshold for how similar the cell in the input data set is to the reference data set. By default, this is 0.4. This is a Pearson correlation coefficient. You can lower this if you want to classify all cells to the reference. And then there are analysis options, filters for differential expression. So by default, it's a 1.5-fold filter and a p of less than 0.05. You typically will want to set an adjusted p-value versus a non-adjusted p-value. This is using a Benjamini-Hochberg adjustment on top of an empirical Bayes lemma-based moderated t-test. With those in hand, you don't need to worry about those other options here. Those are for other separate analyses. You simply can say continue, and the program again will take either several minutes to run to up to an hour, depending on the size of the data set. The data is typically not stored in memory, so you can analyze very large data sets of tens or hundreds of thousands of cells in principle relative to any reference. So again, this will take several minutes to run, so we'll come back in a moment. cellHarmony has completed running. You'll see a new window pop up that's actually has the name cellHarmony, along with a series of results that are produced by the software. Once finished, this continue button will come up. You can simply say OK and quit the program. All the results are saved to this directory. So what is in this directory? By the way, the cellHarmony directory is stored to the same directory in which your expression input file is found. In this case, is the AML file. And there's a number of text file results along with a number of output plots. The first plot that you should look at is in data plots, and it has the extension reordered in it. And so if you open up this reordered heat map, you're going to see what looked like the original alt-analyzed reference heat map produced by ICGS, along with these black ticks at the top. The black ticks indicate actually cells from the sample with cells you're classifying, in this case the AML. So most of the AML cells are actually aligned to this first cluster. This first cluster happened to be the predicted dendritic cells that we originally saw annotated. And there are cells actually from the AML that are actually interspersed in most all the populations that we see. If you look at the cell frequency PDF, it tells you the exact number of cells that align to each and every cell population. The text file that corresponds to that has some additional information within it. This includes p-value, the frequencies for each of these cell populations, along with a p-value of the relative difference of this population in terms of its frequency relative between the classified sample and the reference data set. So in this case, cluster 1 had the smallest p-value times 10 to the negative 20th. Again, these are the dendritic cells, which appear to be overrepresented within the AML data set. As I mentioned, there are a number of other graphical and tabular outputs. The most important tabular outputs are actually the differential expression results. Each one of these files, in this case this cluster 1 again, is the dendritic cells. We can look to see which genes are differentially expressed in the AML versus the reference. So in this case, XCR1 was the most highly upregulated gene in the AML. This is 8.58 log 2 fold, so highly upregulated relative to wild type, along with a number of other genes that are both up and down regulated. If you now actually look back to this data plots folder, you'll see a UMAP projection which shows which cells in blue are associated with the reference cells in red along this projection. You can actually revisualize this UMAP with different groups from the that are assigned from the cellHarmony analysis, since each cell from the classified data set now has a label that's similar to the reference. And you can actually look at the differentially expressed genes themselves in the form of a heat map, a ranked heat map. In this case, the first cluster again is the dendritic cells. The genes in in yellow are upregulated, and the genes in blue are downregulated. And for this cluster of genes, you can see the pathways that are most statistically enriched based on a gene ontology enrichment analysis. So the most significant term within this cluster was external side of plasma membrane genes, along with MHC class II complex. So you can see, importantly, that within each of these different cell states, within the AML, a different set of genes are either upregulated or downregulated, with most of these cell states indicating that more genes upregulated than downregulated within the AML tumor. And again, you can go back to the original ICGS results and see for the 9th cluster from the left, what is that likely cell type, and what are those cells, and what's the biology that's likely associated them, that's going to correspond to the differentially expressed genes and pathways associated with that gene set. Those are the major outputs of cellHarmony. More detail on the different outputs of this program are actually present in the cellHarmony documentation, which again can be found here, and the cellHarmony README that's present within the demo data. If you have additional questions, feel free to contact the AltAnalyze help desk, and thank you for watching.